then its taste may vary and it may taste bad. And that of potassium concentration varied from 3 ppm to as high as 444 ppm. All the range were found in permissible range of World Health Organization, and so the groundwater samples may be treated as potable and can be used for drinking purpose. The references. Thank you. And any questions?
Hemant, I have one question for you.
Yes, ma'am.
What are the main techniques that you use?
क्या मैम? Pardon?
What are the main techniques that you use for sampling or analyze?
Ma'am, samples were collected in pre-washed plastic bottles.
I'm not asking for collection. The technique
Yes.
that is used to detect the parameters.
Pardon, ma'am?
आपने कौन सी टेक्निक यूज की है जिसके आप जैसे आपने पोटेशियम बताया, पोटेशियम
Yes,
आपने
ma'am.
किस टेक्निक से निकाला?
मेथोडोलॉजी मैम ये हमने निकाला है फ्लेम फोटोमीटर से.
फ्लेम फोटोमीटर से. कितना
Yes,
पीपीएम
ma'am.
आया आपका?
मेरा मैम पोटाशियम आया है जैसे 3 ppm टू 444 ppm वाइड रेंज थी मैम.
दिस इज द रेंज,
यस
एवरेज
मैम.
रेंज.
एक ही सैंपल था मैम जो.
How many samples you collected for this study?
इन टोटल 30 सैंपल्स मैम.
Total 30 samples.
यस मैम.
ये सैंपल हमें कलेक्ट करना कम नहीं है? सैंपल साइज आपका कम नहीं है? At PhD level the sample size is too low.
इन टोटल तो 60 है मैम, लेकिन इस पेपर में 30 डाले हैं अभी.
And what are the other parameters?
मैम अदर पैरामीटर्स.
आप मुझे मेन पैरामीटर दोबारा बताइए, क्या क्या किया आपने?
इलेक्ट्रिकल कंडक्टेंस, पीएच मीटर
मेटल
और
बताइए. मेटल.
सोडियम एंड पोटाशियम की डाली है मैम इसमें.
कोई हैवी मेटल नहीं निकाला?
हैवी मेटल मैम वैसे पीएचडी में साइनोप्सिस में तो जिंक लिया हुआ है, लेकिन इसमें नहीं डाला हुआ इस स्टडी में.
ओके. एंड आपने जो पोटेशियम में सोडियम लिया हुआ है वो क्यों लिया हुआ है?
मैम कई सारे एलिमेंट्स लिए थे तो उनमें सोडियम और पोटाशियम भी लिया था.
आपने कॉपर क्यों नहीं ले लिया?
कॉपर मैम शायद पाया नहीं जाता नॉर्मली ग्राउंड वाटर में.
ऐसा कैसे बोल दिया आपने? जब तक एनालाइज नहीं करेंगे तो कैसे पता चलेगा?
जी मैम.
चलिए, थैंक यू सो मच.
थैंक यू मैम. थैंक यू ऑल.
Next one is one. Sarita. Sarita add है? Sarita. Sarita होगी Sarita.
आया तो कौन सा रेड वो मिक्स करना कौन
है ना Sarita? ठीक है.
ठीक है तो
So I would like to invite Sarita from the chemistry department. Her topic of presentation entitled Analysis of Physiochemical Parameters and Heavy Metal Contamination in Agricultural Soil around Brick Kiln. Sarita, can you hear me?
Yes, ma'am.
Okay, please display your slides.
Okay, ma'am.
सरिता ये कहाँ ये यही है
ये नहीं
क्या
है,
कर रही है? सभी ताला ही है।
सरिता है,
अरे फिलिप्स का था।
ये है, ये है।
Ma'am, my screen is visible.
Ma'am, just please wait for 2 minutes. Ma'am, we are presenting your slides from our side, so please just give us 2 minutes.
Okay, ma'am.
So, ma'am, can
Can
you
I start, ma'am?
see your slides? Is it visible to you?
Yes, ma'am. Yes, ma'am.
Okay, so you can start your presentation.
Okay, great. Thank you. Firstly, good afternoon to all of you present here. I, Sarita, research scholar at OSGU. Today, our topic of research is analysis of physico-chemical parameters and heavy metal contamination in agricultural soil around brick kilns. Mainly soil, like air and water, soil is an essential natural resources for human and all living things. Human as well as all the living things use soil in day-to-day life for their requirements and food purpose. For the developing country, industrialization is one of the key factors, but it stimulates the process of environment pollution. In today's world, uncontrolled operation of brick kiln is also contributing to environment pollution, and mainly in contributing environment pollution, air pollution and soil pollution from brick kiln emission is common, which directly affect the surrounding environment. Many gaseous pollutants and ash emitted from the brick kiln reduces the soil quality, which directly affect the human health and plant growth. Ma'am, please next slide. Main objective of this study is to determine the impact of brick kilns on soil physicochemical parameters such as pH, electrical conductivity, moisture content in agriculture area around the brick kiln. The present study also identify within the physicochemical parameters heavy metal contaminant such as copper and chromium in the soil nearby brick kiln. The outcome of this study will be beneficial for the farmers and helpful for the people who want to work in agriculture field. Next slide, ma'am. Main material and methods used for this study. For the determination of the impact of brick kiln on agriculture land, the brick kiln surrounded by agriculture field was selected in the village Chuli Bigadiyan that was situated in the Hisar district of Haryana. Samples were collected in the month of April 2023 from the depth 0 to 15 centimeter and 15 to 30 centimeter at a distance of 5 meter, 30 meter and 50 meter apart from selected brick kiln. All the collected soil samples were labeled properly as 5 meter distance and 0 to 15 centimeter at BKC. BKC means brick kiln, Chuli Bagrian short form as 1, 5 meter 15 to 30, BKC 2, BKC 3, BKC 4, BKC 5 and BKC 6 as so on. After the labeling, soil samples were analyzed. Next slide, ma'am. For the analytical method were used for the pH digital pH meter and for the conductance conductometer was used and heavy metal such as lead, chromium was selected in the soil sample was detected by atomic absorption spectrophotometer. Next slide, ma'am. The pH of the selected soil sample were ranged from 8.06 to 8.32. The soil of the selected area was slightly alkaline in nature, and it also varies with the distance as we move far apart from the brick kiln. pH at the surface level was low, but at the depth 15 to 30 centimeter was slightly high as compared to the surface. Next slide, ma'am. Electrical conductivity was of the selected samples were also less than 4 decisiemon, which was safe for plant growth, and it also varies with the distance as we move far apart from the brick kilns. pH and electrical conductivity both varies with the distance as well as depth. Its moisture content for the selected soil sample was very less than the safe limit. Next slide, ma'am, please. It was only from 0.38 to 0.86%, which is very less and not safe for the plant growth. Next slide, ma'am. After that, heavy metal content lead. According to Dutch standard, the maximum allowable concentration of lead in uncontaminated soil is 85 milligram per kilogram. In the study area, the amount of the lead was in the safe limit. It varied from 18.33 milligram per kilogram to 68.4 milligram per kilogram. Next slide, ma'am, please. But lead concentration varies with the distance. Chromium was also in the safe limit according to Dutch standard. It varies in the studied area from 17.28 milligram per kilogram to 67.5 milligram per kilogram. Chromium and lead both varies with the distance as we move from the brick kiln as well as from surface 0 to 15 centimeter and depth 15 to 30 centimeter. Next slide, ma'am. This is the graph showing the concentration of both lead and chromium in the soil sample from an agriculture area selected around brick kilns. Next slide, please. The next conclusion of this study is that of around the brick kiln, the agricultural soil have pH range slightly alkaline. Electrical conductivity was within the safe limit. Moisture content of the study was very less and not safe for the good crop yield. But heavy metal
Ma'am,
content,
please
lead
hand
and
the
chromium
floor.
was
We have only
within
1 minute
the
left for your presentation.
Okay, ma'am, I will sum up. It means the selected soil was not metal contaminated, but the physical parameters such as moisture content was very less, and all the physical parameters and metal concentration were not uniform, rather varies with the distance from brick kiln. It may be due to the effect of brick kiln. This indicates that the quality of soil is affected by the operation of brick kilns. Thank you, ma'am.
Thank you so much, ma'am, for your presentation. If our chairpersons have some questions, so they can ask.
Sarita, why you collected samples only in the month of April?
Yeah.
सर दैट इज आई डिसाइडेड जो क्रॉप एक ड्यूरेशन होता है कि बिफोर फसल उगाने से जस्ट पहले अप्रैल का मंथ जब खेत खाली होते हैं और मॉनसून से पहले का एंड सेकंड आई माय रिसर्च आई आल्सो चूज द इन द मंथ ऑफ अक्टूबर नवंबर दैट पूरा एक यील्ड एक ड्यूरेशन है वो कंप्लीट होने के बाद आफ्टर मॉनसून
तो आप अक्टूबर नवंबर में फिर से सैंपल कलेक्ट
क्या
करेंगे?
हाँ जी सर
ओके
सर वो हो चुका है ये स्टडी मेरी अप्रैल की रिसर्च
okay so can you tell me why the soil is alkaline at the site bricklane site? What was the reason for alkalinity?
Alkaline may be due to emission from the brick kilns. Emissions mainly rubbers जो होते हैं, rubber batteries ये जलाई जाती है, उसमें हो सकता है मे बी salt हो या फिर ऐसे emission ash में कुछ ऐसे कंटेंट जैसे अगर रेनिंग सीजन में जो पानी है कंटेंट है एश का वो नीचे सॉइल सरफेस पे आके उसके pH को अफेक्ट कर रहा हो.
सैंपल कलेक्शन की आपने सिंगल साइट ली है, व्हाई डोंट यू गो फॉर अदर साइट्स?
No sir, my topic research is industrialization, in which I concluded brick kilns, dumping stations, cotton mills, industrial area, others also collected.
No, you can see this kind of sites in other locations also, no? So you can collect the samples, you can do the comparative study.
Okay, sir. I compare only the WHO limits and other permissible limits, sir, for uncontaminated soil.
Thank you, ma'am. So you can leave now. So I would like to invite now Dr. Sabita from the chemistry department. Her topic of presentation entitled Thermal Behavior of the Polymer Resins and Resins Copolymers. Dr. B. Sabita, can you hear me?
Yes, ma'am.
Okay, ma'am. You can start your presentation. We have 8 minutes timing for your presentation, and 2 minutes are given for the question answering round by the judges.
हमने कर दिया ये भी.
Okay, ma'am, your slides here.
Good afternoon, ma'am.
Can you see your slides?
Yes, ma'am.
Okay, ma'am, you can start your presentation.
My topic is about thermal behavior of the polymers, resins, and resin copolymers. So, in this topic, we are discussing about the content. In this content, introduction, experiments, thermal studies.
Interrupting you in between, please open your video.
Okay, ma'am.
Cannot see you.
Ma'am?
Open your front camera. We cannot see you.
Okay, ma'am. Now it is okay, ma'am.
Yes, ma'am. We can see you now.
Thank you, ma'am. So here in this topic, we are discussing about the high heat resistant polymers. So these are employed from the high temperature and high altitude applications. So here some of the examples of the heat resistant polymers are polyethylene, polytetrafluoroethylene, polycarbonate, polyethylene terephthalate, polyimide. Here, the two types of heat resistant polymers are there, aromatic as well as aliphatic. So here the aromatic groups containing polymers are having the good thermal stability when comparing with the aliphatic. So here some of the previous studies are there. According to this, the 5-5 he can investigate the thermal degradation mechanism on curd phenolic resins by using the NMR spectroscopy. Kondaya, he can report it and synthesis, characterization and the biological activity as well as its behavior of novel acetophenone shift base.
Ma'am, please
Noel-
skip these parts. Please come to the result and discussions and methodology part.
Okay, ma'am. So here, My topic is regarding the two types of polymers. Those are DHPP-FM and DHPP-FM-PMAP. For this, we can discuss about the TGA and DTA. So these polymers are synthesized as well as these thermal studies, TGA and DTA. It is uh, it the graphs can be carried by the Central Drug Research Institute. It is at Lucknow. So these are the two polymers. 2,4-DHPP-FM. It is 1-2,4-dihydroxyphenyl, 2-phenyl ethanone Then 2,4-DHPPE-FM PMAP is nothing but 1-2,4-dihydroxyphenyl, 2-phenyl ethanone formaldehyde para-methoxyacetophenone. So we are discussing about these polymers TGA and DTA curves. So here in this we are discussing about the two polymers, those TGA as well as the DTA. Next slide, it is belongs to the that is figure 1. So in this figure 1 and 2, we are represent the TGA and DTA curves of the this DHPP-FM as well as the DHPP-FM PMAP. So in this study, sir, we are according to this graph for DHPP-FM, the first stage it is the thermal decomposition. It is begins at 270 degrees centigrade and it is ends at the 800 degrees centigrade. But for DHPP FM PMAP, so the value should be 250 degrees. It is the starting stage and 600 it is the ending stage. It is the second stage. It is explained in this figure. So look at this. This is the figure 1. So this figure 1, it is represent the data of TGA and DTA of the DHPP FM. So according to TGA data, so this curve, the decomposition, it is started at 270 degrees centigrade at the first stage, and the second stage it should be started at 800 degrees centigrade. And from this data, so this is the substance component of 2,4-DHPP FM-PMAP. So the decomposition, it should be started at 250 degrees centigrade is the first stage, and the second stage should be 600 degrees centigrade. So by using this
Ma'am,
data,
sorry
we
for the
can
interruption.
explain the
We have
decomposition.
little less time, so can you go come to the conclusion directly?
Okay, ma'am. So here, by using the decomposition data, so this is the decomposition data of the two molecules at a particular temperature, how the weight loss should be taken, and according to this weight loss, we can draw a graph in between the we can draw the graph at the temperature inverse as well as the logarithmic weight of the ratio. It is studies in this figure.
Hello.
Yes, ma'am. Yes, ma'am. Hello.
Savita.
Yes, ma'am. Hello, ma'am.
Hello.
Yes, ma'am.
Savita, can you come to the conclusion slide? Directly
Ma'am?
come to the conclusion.
I'm not audible, ma'am.
Did you prepare conclusion slide? Slide for
Okay,
conclusion.
ma'am. Okay. Okay.
Slide, last slide. Last slide.
So this is the conclusion. According to the thermal data, TGA and DTA curves, when comparing with these two components, the 2,4-DHPP FMPME AP, it is the more thermal stable component when comparing with the DHPP FM. And it is indicated, it is explained by using the oxygen index values also. Thank you.
Okay, thank you, ma'am, for your informative presentation. Thank you
Thank
so
you, ma'am.
much.
Thank you.
हाँ ये मैडम का है ठीक है मैडम का नाम उनको ऐड करो ताकि इन्हें पता चले
So our next presenter is Dr. Sahala Tanvi from the chemistry department. Her topic of presentation entitled Recent Advancement in Wastewater Treatment Technologies.
Hello, ma'am.
Can you hear me? Am I
Yes, ma'am.
audible to you? Okay, sir, please start
Yes,
your
ma'am.
presentation. Please display your slides.
Yes, ma'am. Good afternoon to all of you. I am Nadimullah, Department of Chemistry. I am presenting on the behalf of Dr. Salah Tanveer. She is not well, so I am presenting. I am co-author of this presentation. My topic is recent advancement in wastewater treatment technologies. Ma'am, next slide, please.
Sir, please open your
Introduction of wastewater.
camera
Okay, ma'am.
so we can see you.
Yes,
Yeah,
ma'am.
fine now, it's fine.
Ma'am, next slide, please. Introduction of wastewater treatment technology. Wastewater treatment is process which remove and eliminate the contaminant from the wastewater and converted this into all the influent that can be returned to the water cycle. The treatment process takes place in water treatment plants. There are several kinds of wastewater which is treated at the appropriate types of wastewater treatment plant. For domestic wastewater, the treatment plant is called as sewage treatment. For industrial wastewater treatment, either takes place in the separation industrial water treatment or a sewage treatment plant. For the types of wastewater treatment plants include agricultural wastewater treatment and leach treatment plants. Ma'am, next slide, please. There are traditional wastewater treatment. Traditional wastewater treatment comprises primarily primary, secondary, and tertiary procedure that based on biological, chemical, and physical treatment process. The most common biological process is sewage water treatment. Is a treatment which active slash traditional wastewater treatment facility conduct the active select process or additional form of biological treatment such as biofiltration. These processes have been able to remove various pharmaceutical range from 20 to more than 19%. Ma'am, next slide, please. Traditional wastewater treatment can be divided into three categories: physical, chemical, and biological category. And these are three categories subdivided into the physical, divided into the membrane and reverse osmosis or sedimentation, or coagulation, and chemical process is subdivided into the ion exchanger or chemical oxidation, chemical geolytes, etc. And biological wastewater treatment can be divided into biodegradable and non-biodegradable. Biodegradable are active slash enzyme bioreactor and oxidation pond membrane bioreactor. Ma'am, next slide, please. Physical method of wastewater treatment, in which physical forces are utilized to remove contaminants, were among the first wastewater treatment technology used. The method are typically employed when water is heavily polluted. Most often, physical water treatment methods such as screen sedimentation and steaming are used to remove the solids. No chemical are involved in this process. One of the main temperature of physical wastewater treatment includes sedimentation, which is a process of suspended in the insoluble having particles from the wastewater. Sedimentation and gravity saturation, membrane technology of wastewater treatment, floating coagulation. Ma'am, next slide, please. Sedimentation and gravity separation. The process of hollow particles is a suspense in water to settle on the surface under the effect of gravity. The particle that settle out from the surface become sediment, and in water treatment is known as sludge. Membrane technology involved in several steps: microfiltration, ultrafiltration, nanofiltration, reverse filtration. Ma'am, next slide. The most used physical treatment is coagulation and reverse osmosis, that is RO. And coagulation, non-settled solids are allowed to settle when suspended solids do not settle down through the sedimentous or gravity. Coagulation is the term for this process. This is possible to employ alum, starch, ferrous mineral, ammonium salt, and active silica. Coagulation made of non-ionic polymers, anionic polymers, non-ionic polymers and synthetic cationic polymers are also effective, but they are usually more expensive than natural coagulation. The most essential gourmet parameter in the coagulation process are temperature, pH, and content time. Specific coagulation are added to biological treatment unit to remove the bacteria and other organisms that may be floating in the water. It is most significant part of the water treatment unit and it is used to a variety of purposes, include wastewater treatment, recycle, and pollution removal. Ma'am, next slide. First of all, we added the coagulate in the with the impurity of water, and then after the coagulate trapped and from the precipitation. After then the precipitation trap into the impurity of settle down on the water, and we can easily remove the water. Ma'am, next slide. Chemical method or chemical treatment is a term that refer to an number of different ways to treating wastewater and the chemical used to water treatment from the method to method. For example, sodium hydrochloride acid, and magnesium hydroxide are used to different chemical treatment. There are many specific ways to using the chemical treatment: water, ion exchange, and chemical stabilization, chemical precipitation, geolite, etc., and other types of method used in the chemical method. The next slide. In the ion exchanger, a solid substance exchanges the hazardous ions of the wastewater for non-toxic ions. There are two types of ion exchanger: anionic and cationic exchanger, which can exchange anion and cation respectively. Ion
Sorry,
exchange.
sir. Sorry for the interruption. You have only 1 minute left, so you can conclude. You can directly come to the
Ma'am,
conclusion.
ma'am, you can say the calculation.
Please come to the conclusion point.
Okay, ma'am. Then the conclusion, the treatment of wastewater crucial is affecting the environmental due to the increased urbanization and sterilization. Wastewater generally and treatment have become a grow concern in the 21st century. Many wastewater treatment options are employed to address the problem to growing the environment pollution, include physical, chemical, and biological, primary, secondary, and tertiary technology. The employment of some treatment strategy has the potential to produce secondary contaminant. Advancing wastewater recycle have made it possible to produce water to virtually and quality. Water recovery system incorporate a variety of safety precautions to reduce the environmental risk associated with the various rescue applications. Under the present condition, improve or integrate wastewater treatment technology are critical required to ensure high quality water, reduce chemical and biological pollutants that enhance industrial production operation, integrate the approach which might overcome the limited the single treatment technology seem to be a viable option for the efficient wastewater management. Thank you, ma'am.
Thank you, sir, for this informative presentation. Our judges have few questions for you related to your presentation.
Yes, ma'am.
Hello.
Yes, ma'am.
So can you tell us any coagulant,
जी मैम.
name of any coagulant?
Ma'am, I am not understanding.
Do you know the meaning coagulant? Coagulation?
Ma'am, coagulation.
Coagulation.
Coagulation. Ma'am, the coagulation is the process, the physical process in which not involved in chemical reaction. That is the that is then the some impurities in water involved that they coagulate and some
Alum a coagulant or not?
alum is coagulant, starch, alum, and ferrous mineral, aluminum salt. That are alums, are coagulant.
Okay. So as per your study, which is the most efficient technique to treat wastewater?
Most traditional techniques in wastewater is microbial fuels wastewater treatment, oxidation pond, or RO and membrane filtration RO, and the natural process that are involved in the most recently advanced and research in the technologies.
Okay, thank you.
Thank you, ma'am.
Thank you, sir. You can leave
Thank
the
you, ma'am.
presentation.
Can, can we leave?
Yes, you can leave.
है ना पारुल है तो उसके उसमें करनी
So I would like to invite our next presentee, Ms. Parul from the Chemistry Department. Her topic of presentation entitled A Comparative Study on Photocatalytic Activities of Various Transition Metal Oxides Nanoparticles Synthesized by Wet Chemicals.
पारुल से करी पारुल हाँ डिस्प्ले करेंगे सारी
So now we will be displaying these slides. You can start your presentation. Ma'am, we have 8 minutes for the presentation and 2 minutes are for the question answering round that will be done by our judges. So within 8 minutes you can conclude. Thank you, ma'am.
Good afternoon, everyone. So, I am going to present the topic comparative study on the photovoltaic activities of various transition metals and particles.
Ma'am, we cannot hear you. Miss Parul, can you hear me? Am I audible to you? Ma'am, we cannot hear you. Please unmute.
How can I increase the volume?
अब आ रही है
Ma'am, please, can you hear me?
Ma'am, can you hear me?
Yes, we can hear you.
Thank you, ma'am. So, good afternoon, everyone.
Ma'am, please be little louder.
यहाँ से
Ma'am, please be little louder because we cannot hear you properly. And it is not audible to the audience also.
Good afternoon, everyone. Today I am going to present on the topic comparative study on the photometric activities of various transition metal oxides, nanoparticles synthesized by Soldier and wet chemical method.
Ma'am, your voice is not at all clear.
Ma'am, I don't know what to do.
हम नेक्स्ट कर रहे हैं आपको उसकी वॉइस बिल्कुल भी क्लियर नहीं है बोल दिया ना
Ma'am, वो तो मेरा
उनको मैसेज पोस्ट हो रहा है वॉइस इज नॉट क्लियर वी कैन क्या करें नेक्स्ट करें फोन पर डालें नेक्स्ट को भेज दीजिए नेक्स्ट है इसका फिर लास्ट में देखते हैं है ना इसका नेक्स्ट पे अपन करते हैं यहाँ पे निर्वाणी करते होगी कोई निर्वाणी है ना इसको होल्ड पर डाल दीजिए
So I would like to invite our next presentee, Nirvani Bharti from the chemistry department. Her topic of presentation is synthesis and spectral identification of semi-carbazone thiosemi. Ma'am, can you hear me?
Good afternoon, ma'am. Can you hear me?
Yes, ma'am.
Okay.
Please display your slides, ma'am.
Okay, ma'am.
Ma'am, you have 8 minutes. In 8 minutes, you have to wind up your presentation, and 2 minutes will be given for the question answering round. That will be done by our judges. So, thank you, ma'am.
Okay,
You can
ma'am.
start your presentation.
Synthesis and spectral identification of semicarbazone, thiosemicarbazone derivative having transition metal complexes. It is our view. There are some our view. Next is introduction. Free radical is an atom or molecule that bears an unpaired electron and is extremely reactive, capable of engaging in rapid change reaction that destabilize other molecules and generate many free radicals. In animals, these free radicals are deactivated by the antioxidant. These antioxidants act as an inhibitor of the process of oxidation even at relatively small concentration and thus have diverse physiological role in the body. The body is constantly exposed to the negative and sometimes lethal effects of oxidants during normal physiological process. The harmful free radicals such as hydroxyl, peroxyl, and the superoxide anions are constantly being produced as a result of in the living system. On a daily basis, up to 5% inhaled oxygen may be converted to reactive oxygen species. These ROS have the ability to bind to cellular structures and have been implicated in a number of pathological processes such as aging, inflammation, reoxygenation of arsenic tissues, arthrolysis, cancer, and even Parkinson's diseases. In man, collaborative efforts by the multidisciplinary teams from various national and international agencies to be coordinated and managed at different elections. Two processes which produce free radicals in vivo has been identified as the name conventional antioxidants have been shown to improve animal performance during conditions characterized by increased tissue oxidant level such as stress, injury, and the infections. The semicarbazone is an electron withdrawing group and exhibited antioxidant activity. Favorable substituents may increase their scavenging effect. Semicarbazones and thiosemicarbazones are important compounds obtained by condensation of semicarbazides with suitable aldehydes or ketone. Thiosemicarbazones is a derivative of semicarbazone which contains sulfur atom in place of oxygen. The semicarbazones and semicarbazones usually behave as a chelating ligand, usually reacts with the metallic cations giving complexes. The synthesis of transition metal complex with semicarbazones and thiosemicarbazones ligands are due to their pharmacological properties. Considering the biological potential of semicarbazone, thiosemicarbazones, and their derivatives of transition metal complexes, herein the synthesis route of some of these derivatives is reviewed for the antibacterial, antifungal, antimalarial,
Uh,
antineoplastic, and
Sorry for the
antiviral
interruption,
activity.
ma'am. Sorry for the interruption.
Yes, ma'am.
Can you proceed to the next slide that includes methodology or result or discussion?
Yes, ma'am.
Can you move to the next slides that include methodology or result and discussions?
Ma'am, your voice is not clear.
Ma'am,
Some
can you hear me now?
disturbance occur.
Please move to the slide methodology. Next slide, methodology.
Yes, ma'am.
Yes, ma'am, please move to that slide because we are running short time.
Okay. The synthesis of semicarbazones was carried out using the
Ma'am,
treating urea with
ma'am,
hydrogen.
sorry for the interruption. You can proceed with your back slides, which include introduction part, I guess. You can start from there also.
Okay, ma'am.
Ma'am, you can start from here.
Spectral analysis of semicarbazone. Chemical bonding nature of semicarbazone and thiosemicarbazone with transition metals. The raw material for the synthesis of semicarbazone and thiosemicarbazones is semicarbazide. The compounds prepared by treating urea with the hydrogen and the chemical reaction as semicarbazone, thiosemicarbazones are a derivative of an aldehyde or ketone formed by a condensation reaction between a ketone or aldehyde of semicarbazide and thiosemic.
Next slide.
The coordination mode of semicarbazone is very sensitive towards the minor variation in the experimental conditions. They are very versatile ligand which can coordinate to the metal as neutral molecules or after deprotonation as anionic ligand and can adopt a variety of different coordination modes. Thiosemicarbazones and the semicarbazones act as ligands because they have better coordination tendency. And both tautomeric forms have an efficient electron delocalization along the semicarbazone moiety. Aromatic substituents on semicarbazone skeleton can further enhances the delocalization of electron charge density. The classes of compounds usually reacts with the metallic cation, giving complexes in which the semicarbazone behave as a chelating ligand. Upon coordination to a metal center, the delocalization is further increased through the metal chelate rings. The coordination of carbazide are further increased if the additional donor atom in most of the semicarbazone were synthesized as tridentate ligand and in some of the cases semicarbazone. This is the UV VIS spectral analysis of semicarbazone. The UV-VIS spectrum of the ethanolic solution of semicarbazone recorded in the wavelength region from 200 to 600 nanometer is represented in figure. The spectrum shows two absorption bands due to pi star transition at lambda max equal to 200 nanometer, 220 nanometer, and the lambda max 289 nanometer. The third one at lambda max is equal to 316 nanometer due to N to pi transition in the synthesized compound. These transitions are the characteristics for valerian semicarbazone as a skiff base. The spectrum shows no transition above 450 nanometer, which makes vanillin semicarbazone convenient for many non-linear optical applications. NMR spectral analysis of semicarbazone. The NMR spectra conclusion, the synthesis of semicarbazone was carried out using the treating urea with hydrogen of semicarbazide in standard solvents. Methanol, thiosemicarbazone metal complexes have also been investigated as radical squenchers. Skiff-based metal complexes will were also found to be active against yeast and also act as herbicide, insecticide, rodenticides, plant growth regulator, and their uses in the treatment of diseases such as anti-tumor, epilepsy, mental disorders, and their enhanced biological activity has been an active area of the investigation for the future researchers. The composition of ligands can be determined by the CHN analysis. For the characterization of these compounds, 1H NMR and IR spectral studies are more important. Thank you.
Thank you so much for your presentation. Judges have few questions regarding your presentation. So I invite our judges to have a question round with you.
Hello, Nirvani.
Yes, ma'am.
Nirvani, I have only one question for you. Which
Yes,
type of
ma'am.
transfer you have reported for semicarbazone? The UV
Yes, ma'am.
spectroscopy. By UV spectroscopy, which type of transition you have reported?
Yes, ma'am. Pi to pi transition and n to pi star transition.
Okay, thank you.
Pi to pi star transition at lambda max 220 and 289 nanometer. And the second है n to pi transition lambda max is equal to 316 nanometer.
Thank you, ma'am. Thank you for your presentation. You can leave.
आगे को ऐड करो फिर बोलूंगा कनेक्ट नहीं अब वही ना ये आके दिए ये है तो
ये तो इसकी वो प्रेजेंटेशन है और खुद नहीं है प्रेजेंटेशन अपने कर रखी है ये रही हो तो कुछ
आप प्रेजेंट करोगे प्रेजेंटेशन स्लाइड आप प्रेजेंट करोगे कि वो करेगी
मैं करूँगा
आप करोगे
अब आ गया और हो गया
Okay. So I would like to invite Ms. Parul. She is from the Chemistry Department, and her topic of presentation is entitled "A Comparative Study on Photocatalytic Activities of Various Transition Metal Oxides, Nanoparticles Synthesized by Wet Chemistry." Ma'am, can you hear me now?
Yes, ma'am. Ma'am, my voice is coming.
Yeah, your voice is audible. We can hear your voice. So,
Thank you,
ma'am,
ma'am.
you have 8 minutes timing for your presentation. You have to wind up your presentation within 8 minutes, and 2 minutes will be given for the question answering round that will be done by our judges. So, you can
Thank you.
start your slides.
Okay, thank you, ma'am. Ma'am, how can I change my slides?
Ma'am, don't worry, we have a person. He will be changing the slides.
Ma'am. Okay, ma'am. Good afternoon, everyone. Today I will be presenting on the topic comparative study on photoelectric activities of various transition metal oxides, nanoparticles synthesized by sol gel method and wet chemical method. So, in this presentation, I will be. Can you please change the slide? Introduction. So, photocatalysis is the process that uses light irradiation to activate catalyst in order to initiate a chemical reaction. This process aids in degradation of organic pollutants on daily basis and is beneficial for wastewater treatment system. Industrial effluent produces dyes such as textiles, ceramics, paints, and pulps and papers industries which multiply the level of toxicity in water streams. Semiconductor photocatalysis such as zinc oxide, titanium dioxide, and iron oxide have been widely applied for their functionality on the photodegradation. Oxide-based materials like the zinc oxide possesses a large and direct band gap, thus requiring a massive amount of energy of incident beam photons to generate the photoreaction. And this contributes in three ways. This the photocatalyst, it narrows the band gap and it promotes the conductivity of Zdeno and mobility of charge carriers. Also, the band gap between valence band and conduction band, it reduces. Next slide. Basically, the process of photocatalysis takes place when energy is higher than the source in comparison to the energy gap of a semiconductor, which is Zdeno in this study. Thus, electron excited from the valence band jumps to the conduction band to form possibly hole and positively hole an electron on the surface of Zdeno. Like I have typed the equations here. The mechanism, the zinc oxide, then it takes in presence of sunlight, it releases an electron to the conduction band. So these electron hole pairs involve in a redox reaction as shown in the equation 3.7 and 3.8, producing hydroxyl radicals. The dopants, the silver and nickel, serve as the electron scavenger, equations 3.9 and 3.10, to trap excited electron that intercepts the recombination between the pair and the photo-generated holes, thus resulting in increased lifespan of excited electrons. This gives more time for both excited electron and the holes to react with H2O and generate exceptional oxidant species, that is free radical O2 negative, NH2O2, which later degrades and the dies. That equations 3.111 and 3.14. The oxidant species is non-selective and is highly reactive that has ability to degrade pollutants by breaking down organic bonds, including destroying the aromatic rings also of the dye molecules. Equation 3.49. The generating the more oxidant species enhances the photocatalytic performance. The possible mechanism I have mentioned here. So please next slide. The photo uh, in this study I am mainly comparing the photolytic activity of the these transition metal compounds. The photoelectrical activity of the silver and nickel is a nano. Means I have taken zinc oxide as a nanoparticle and doped with silver and nickel respectively, and then I have measured by determining the decomposition of methyl orange on each interval at constant room temperature. The catalyst loading was 100 milligram of silver and nickel is a nano catalyst in a medium breaker containing that methyl orange solution with some initial concentration. Then the UV visible light was done at 352 wavelength and the 8-watt power. The dye solution was extracted at every 40-minute interval. The photoelectrical analysis were performed by UV visible and absorbance was made. The methyl orange absorption peak was measured as 464 nanometer. The photoelectric efficiency was measured in regard to a maximum photodegradation collected at each interval. Absorption control graph was produced in prior to photodegradation by excluding UV light irradiation. The photocatalytic activities were compared in terms of rate constant, decoloration of dyes, the pH, the contained graphical method, also the log of A naught by 80 against time of the photocatalysis. Next slide. Then the all the dyes and this the nano catalyst I have taken. Also the I have taken 4 catalysts uh, that instead of doping also like titanium dioxide, tin dioxide, and iron oxide. So all these 3 also I have done the study on the dyes like methyl orange to study about the degradation of dyes and also, the comparative studies on zinc oxide and titanium dioxide photocatalysis could have been performed to many and by many other researchers, where synthesis zinc oxide has shown a higher performance than titanium dioxide. Uh, the surface area of photocatalyst is one of the key parameters that affects photocatalytic reactivity. Among the 4 catalysts, the zinc oxide displayed the lowest specific area, while the DIGOSA P25 photocatalyst had the highest specific surface area. The results revealed that despite having a specific surface area, zinc oxide exhibited a higher photocatalytic activity than did the titanium dioxide and tin dioxide. Quantum efficiency may be used to explain the differences in photocatalytic activity. Zinc oxide has a greater quantum efficiency than titanium dioxide and tin dioxide. Also, the critical factor affecting the photocatalytic activity is the band gap. In our study, tin dioxide exhibited a relatively low activity because of the wide band gap, that is 3.64 electron volt. Since a whole band gap photocatalyst needs a large amount of UV radiation to excite electrons and to form electron hole pairs in the catalyst, and while the zinc oxide and titanium dioxide exhibited comparable band gap, that is 3.24 and 3.20 electron volt respectively. Next slide. So I have attached some results of the XRD and some images of the this transition metal oxides. So the image one that is showing the red graphs, it is about XRD pattern that is ranged between 20 degree to 90 degree for silver and nickel doped the nanoparticles. Means ki the zinc oxide is doped with silver and nickel. In the first row, it is doped with silver, and the second half is for nickel. And the good crystallinity was achieved as the different peaks displayed with a good match. And the zinc oxide woodside hexagonal structure has a greater efficiency among all these. For nickel Zardano dog structure, as shown in uh, later half graph, it was revealed that the boundaries of xerenocrystallites, uh, have a single phase of xeranobotrite structure, where the previous has a single type structure. The second,
Ma'am, please proceed to the conclusion slide.
but ma'am,
Ma'am, please proceed to the conclusion slide.
okay, ma'am, actually I have not made a conclusion slide in this. Okay, I have done conclusion on this graphs only.
So, ma'am, can you conclude your entire study in 2 to 3 lines? So the briefing
Yes,
can
ma'am.
be done according to that.
Yes, ma'am. Wait a minute.
Ma'am, so your conclusion will be completely based on your results and discussions. So according to
Yes,
that,
ma'am.
you can make
Yes, ma'am.
conclusions.
Ma'am, examination of photocatalytic efficiency of the transition metal oxides have been carried out at definite time intervals until the regular blue color of the aqueous solution of uh, methylene blue and the red color of methyl orange become colorless. The noticeable point was best photocatalytic capability of iron oxide photocatalyst. Among all the oxides I used, the iron oxide given the best result. FRD analysis displayed very small crystallite size of transition metal oxides. Also, the EDX studies and SEM studies confirmed the morphology and elemental composition of the samples respectively. Also, the BET results revealed that iron oxide exhibit highest surface area and the lowest particle size. The photocrystallites and the particle size and large surface area and wide band gap of the uh, iron oxide and decrease recombination of charges onto surface of photocatalyst. So, in the, on to conclude, the rate was the iron oxide was the highest, then cobalt, then manganese, and then zinc at the last. So, in conclusion, the synthesized nanosized particles, especially iron oxide nanophotocatalyst, could be the auspicious applicant to degrade the waste constituents and has the forthcoming applications in the resolving pollution affairs with the environment. Thank you.
Thank you, ma'am. It was very informative presentation. So I will just have 2 questions for you. Okay, ma'am, we do not have any questions for you. You can leave the slide. You can leave. Thank you so much.
Thank you.
Okay, so I would like to invite our next presenter, Dr. Ram. He is from the zoology department, and his topic of presentation is status of Indian peafowl. So you can start your presentation. Sir, we cannot hear you. Can you hear me? Can you hear me? Sir, we cannot hear you. Your voice is not at all audible to us.
वॉइस आ रही है मैम मेरे पास यस
Okay,
मैम
okay, sir. Okay, sir. You can start your
यस मैम
presentation.
यस मैम today I include some content. First, I discuss introduction, feeding habit and habitats, roosting, nesting tree, factor affecting the population size, reason for population decline. Next, I discuss the in my presentation thing to revive the population, and last I discuss the conclusion and summary. First of all, ma'am, I give Indian peafowl or Pao Christus is an important bird residing near to humans settlement like open scrap and wood
Sorry for
cave.
the interruption. You have
First
only
of
6
all,
minutes for completion of this slide.
1963, 1963 national birds, uh, peacock, scientifically
Sir, the
known as
slide is
the
not moving.
Paocristus. So, first of all, I discuss the feeding habit and habitat. Peacock may, for example, we can say feeding habit, seeds, lizard, snake, pest, and earthworm, and some lizards, feeding habit of the peacock. And habitat, we can discuss the habitat just like a tree canopy. And the birds mostly survive on grass or seed and product like wheat, rice, pea, weed, seeds, etc. Agriculture pest and some small snakes eat the feeding habit of the peacock. And some we can discuss the रोस्टिंग एंड नेस्टिंग मतलब पिकॉक कहाँ पे रह सकता है जैसे फॉर एग्जांपल मैनी ट्रीज मैंगो यूकलिप्टस फेदा एंड Prospect Mangifera indica, Acacia, Ficus religiosa, so many roosting and nesting tree species of the peacock. Next, I discuss the predator of Indian peacock. Which is the predator of Indian peacock? I discuss the jackal, python, dogs, and common Indian monitor lizards are the predators of the Indian peacock. Next, factor affecting the population size. Which factor affecting the population size? Many factor affect the population size. For example, tree cover, scrubland, herb cover, agriculture activities, presence of natural water bodies, human disturbance. इसमें agriculture, for example, sometimes eggs are break by the machine, and sometimes agriculture, or sometimes there is no source of the water, so peacock population also affected. So factor affecting the population of size. Next, reason for population decline. So many reason just for example we can discuss the habitat destruction, lack of natural water bodies, shrinking size of open woodland and tree cover, mechanical farming, pesticide. So pesticide also decrease the peacock population decline. Next, we discuss the step to revive species population. For example, habitat conservation, identification of risky zone, cultivation of foreign crops like maize, pea, protection and plantation of roosting and nesting tree, awareness program. For example, next, economic importance of pea fowl. What is the economic importance of pea fowl? We can discuss the just for example egg, meat, and sometimes reason for population decline, and sometimes feathers are also useful. According we can say Ma Saraswati, Virajman on the peacock. So this is the economic importance of the peacock. And step to last, I discuss the conclusion and summary. Peafowl is an important Indian bird having religious importance. We can say most important cultural value, religious value, economic value. We can say peacock importance value है. This is the conclusion. And birds population seems to decline due to habitat destruction and pesticide application. So more pesticide used in the crop, and sometimes peacock eat the weeds. So peacock population also decline. The
Okay, sir.
population need to revive through various conservation
Sir, can you
practice
hear
like
me? Please
conservation of
proceed with the
woodland
conclusion.
and construction of water bodies.
Sir, please.
Thank,
Yeah.
thank you, ma'am.
Thank you, sir. Thank you for the presentation.
Thank you.
So thank you. You can leave the slide. You can leave.
Next,
ये कौन सा लेना
Vicky.
बेटे नोट कनेक्ट
एक काम करो क्वेश्चन समझे होगा इसमें और करो नहीं
निकिता डागर है।
आप भी बता दो
ये सिर्फ मंजू मंजू निशा मंजू
डॉक्टर
वो
कौशल
तो है मैम, वो है।
फारूल का होगा
फारूक को भी इसमें ना डॉक्टर कौशल संजन
नहीं
अभी थे वो,
अभी वो हट गया निकल गया
कोई है इसमें?
इसमें सिर्फ मंजू और निशा है
निशा और मंजू। मंजू है ना? ठीक है, मंजू है।
कमाल है कौन है
मंजू है ना? सारी डाउनलोड करो फिर मैं बोलूँगी। मंजू है?
ये किस
कौन कौन आ गया इसमें हटा दो फारूक
मंजू है
ये लगा लो क्वेश्चन संजन
मंजू
आ गए ना? इनको आ गए।
हाँ इनको क्वेश्चन
चलो ठीक है।
Okay, so our next presenter is Dr. Kaushlesh Ranjan. He's from Biotechnology Department from the University Sardar Vallabhbhai Patel University, Meerut. His topic of presentation is proteomics and its application in human and animal health.
Okay. So, मैं audible?
Yes, sir.
क्या मुझे
you
सुन
are.
पा रहे हैं?
Yes, sir. Yes, sir. We can hear you. Just give me a 1
Okay,
minute.
okay.
आप साइड पे जाइए, वो करेंगे।
प्रेजेंट मैं कर रहा हूँ सर कोर्स पे कर रहा हूँ प्रेजेंट मैं कर रहा हूँ
My slide is visible there?
No, sir. We are having your slides. We are processing it. It is just taking 2 minutes. Just.
Okay.
Okay, now, yeah. Yes, sir. Can you hear your? Can you see your slides?
Just 1 minute, madam.
Yes, sir. You can start your presentation.
Okay, so I am going to start now.
Yes, sir.
So, good afternoon, one and all. So, I am Dr. Kaushlesh Ranjan. I am working as assistant professor at Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. So, I am going to present a topic that is called proteomics and its application in human and animal
Sorry
health.
for the interruption, sir. Your voice
Okay,
is not clear and it is not your pitch, and it is not also audible.
Okay. I think it's the internet connectivity issue, madam. So, my topic is proteomics and its application in human and animal health.
Sir, can you bring
Now
your
it-
microphone close to your mouth, please?
Okay, ma'am. So my topic is proteomics and its application in human and animal health. Is it okay?
Yes, sir. Yes, sir. It's fine. I
Okay.
can hear you.
Okay. So we know that proteomics, very common topic, which deals with the overall protein diversity of an organism at a particular times. That is all the proteins present in an organisms at a particular point of the time. So in proteomic studies, proteins are isolated and separated from the organisms, then analyzed using various techniques such as SDS polychloride electrophoresis, then Western blotting, mass spectrometric-based similar techniques such as Marley-Tops and other techniques are there. So some of these techniques I will discuss here only in the brief. So they talk technique called the very common technique that we call the SDS page of the proteins. So SDS page is used for the separations of the protein mixtures, for the analysis of the different types of the proteins. So it is based on movement of the proteins in electric fields on the basis of the mass and charges. And here the movement of the proteins is also dependent on the primary structures of the proteins, which can determine using the SDS applications of SDS, then beta-mercaptoethanols or ditheritols, also called the DTT, because the native proteins are present in tertiary and quaternary form of the protein structures, which are not easily movable in the polyacrylamide gel electrophoresis. So the primary structures of the proteins are separated in the polyacrylamide gels, which are made up of acrylamide, bisacrylamide in 29:1 ratio. They also contains ammonium persulfate and timides. Then this system is run with the help of 110 to 120 volts of the DCFT currents, which are used for the electrophoresis of the protein samples. So here we can see that this is the native structures of the proteins. Before applications of the SDS, actually, these proteins exist in the tertiary or quaternary forms. So after applications of the proteins, the hydrogen bondings are broken down and proteins become native form, that is only the primary structures with the negative charge on the entire proteins. And these type of the apparatus are used for the SDS page of the proteins. The protein samples are loaded in the gels. Here we can see here on the gels. Then these gels are kept inside this electrophoresis apparatus. This is a vertical type of the electrophoresis apparatus. And current is applied here. Then protein samples are separated based on the mass and charge ratio. Afterwards, the gels are removed and the gels are stained with the help of massive brilliant blue stain, or also ethylene bromide dyes can be used to identify the specific types of the proteins, and proteins are visualized here as a specific dots, which are visualized in the blue colored dots. The next technique is Western blotting. So again, Western blotting technique is also very common techniques present in many of the labs. So this technique is also referred as protein immunoblotting. So it can identify the specific proteins from a mixture of the protein samples. And Western blotting steps usually includes three major steps. First step is called the size-based separations of the proteins, which is done using the polyacrylamide gel electrophoresis. Then transfer of the proteins, then labeling of the target proteins using primary and secondary antibodies for visualizations of the specific protein samples. So on these slides, we can see the basic layout of the Western blotting techniques. So protein samples are isolated. The samples are treated with SDS. Then the samples are used for the gel electrophoresis. Then transferred to the membranes, nitrocellulose membranes through the blotting techniques. Then primary antibody and secondary antibodies are applied. Then these protein samples are visualized using the specific type of the enzymes, for example, HRPO, horse oxidase, and other biomolecules. Then the third methods of proteomic techniques is called the mass spectrometry. So this mass spectrometry techniques,
Excuse me, sir.
the mass charge
Excuse
of
me.
the molecules in a sample. Hello.
Dr. Kosle,
Yes, sir.
please
Hello.
tell us the applications in humans and animal health only. We know the
Okay,
technique.
sir. Just going there only, sir.
Yes, come
Just
to that
going there
point.
only, sir. Okay. So these are the mass spectrometry techniques. The X-ray crystallography techniques we can also use for the proteomic study. So this is a basic layout of the X-ray crystallography. So the proteomics techniques can be used for the several applications in human and animals. There are several applications, only few applications I have taken here because of the time constraints. So one of the most common technique, most common applications of proteomics is proteomics in human and animal disease diagnosis. So disease diagnosis, proteomics can be used. So proteomics has important role in diagnosis of disease in human and animals. So by analysis of the and comparisons of the protein data with the help of protein data banks, the proteomics can show the molecular signatures associated with the various diseases, that is several pathogens, bacteria, virus, fungus, parasites, prions, and many of genetic diseases. Their molecular signatures or molecular fragments of the proteins can be identified and compared with the help of the protein data banks. For example, tuberculosis and HIV have been identified with the help of the proteomic data in human diseases, and also in case of the animals, the food. foot and mouth disease, then bovine tuberculosis, and several other diseases have already been identified based on the proteomics data. So few examples only I have taken here. Then second application is identifications of foodborne pathogens and their toxins. So proteomic techniques can play a crucial role in detecting the foodborne pathogens. So a study shows that more than 250 different microbes, most of these microbes are bacteria and some of the fungus are referred as foodborne pathogens. They can cause severe disease in humans, even lead to the death also. So several foodborne
Thank you,
pathogens
sir. You answered
such
very
as
well. Thank you so much. We are running with a short of time. That's why I'm interrupting in between. So thank you
okay,
for
okay, only
your
2-3 slides are
informative
there.
presentation.
Then urgent detections, then proteomics in food safety, and conclusion.
Next.
Thank you so much, sir. You can leave.
Okay, thank you. Thank you, thank you.
विक्की है विक्की
Device not connected.
कौन किसका कनेक्टेड है
मंजू
मंजू
वाला।
है ठीक है इसी को करते हैं चलो मंजू ठीक है
So our next presentee is Manju from the mathematics department. Her topic of presentation is a survey on the properties of some power graphs.
A very good morning, a very good evening to all. Am I audible?
Ma'am, just give us 2 minutes, ma'am. Please give us 2 minutes.
I'm Manju presenting on the topic a survey on the properties of some power graphs. As we all know, algebraic graph theory explore the relationship between algebraic structure. Uh, Ma'am, please share my PPT or PDF.
Yes, ma'am. Just give us 2 minutes. It is
Okay,
processing.
ma'am. Okay, ma'am.
Okay, ma'am. We have only 6 minutes for your presentation, so you can wind
Yes,
up your
ma'am.
presentation within 6 minutes, ma'am.
Okay, ma'am. Okay, ma'am.
Okay, ma'am. You can start. Thank you.
Uh, I'm Manju presenting on the topic a survey on the properties of some power graphs. As we all know, algebraic graph theory explore the relationship between algebraic structures and graph theory. One significant area of study within this field is the power graph. In recent years, there has been substantial progress in understanding various aspects of power graph, including their connectivity, spectral properties, isomorphism, automorphism, characterization, and the terms of groups. A power graph is a type of graph derived from a mathematical structure. The power graph G of G is a simple graph where vertices represents the elements of groups and two distinct vertices are adjacent if one is a power of other. In this paper, our main objective is to provide a comprehensive survey focusing on the properties of enhanced power graph, reduced power graphs. These variations of power graph often define insights into underlying group structure and its relationship between the graph theoretical properties. Firstly, the concept of directed power graph introduced by Carville and Kinin, and that is a digraph with vertex G, and for any AB belongs to the group, there is directed edge from A to B in PG if and only if A ki power k is equal to B, where k belongs to natural number. Directed reduced power graph, ma'am, please next slide. Directed reduced power graph is a graph digraph with the vertex G for elements. Two elements belongs to G. There is an arc from two elements between two elements if and only if these U and V are not equal and one is the power of other. And also undirected reduced power graph is underlying graph of the power graph. And the next term is the enhanced power graph. It was introduced by Alupur that includes properties for both power graph and commuting graph, where commuting graph is denoted by C of J. Enhanced power graph, it is a simple graph with the vertex set consisting of all the elements of G where two distinct vertices are adjacent if and only the cyclic group generated by these two elements is the subgroup of G. Next to the study of we are studying here spectral properties of the power graph like L spectra and A spectra. Ma'am, please next slide.
Applications.
Next
Uh,
one.
wait.
Uh,
L spectra. L spectra is the eigenvalues of the Laplacian matrix
Ma'am,
are
is
L
there any
spectrum of the
confusion
graph.
in your slide?
Um, wait.
There is a conclusion slide.
One theorems
Con-
and
Conclusion slide. Ma'am, last conclusion slide. In this paper, we have observed many results obtained by many authors in for the spectrum, vertex connectivity, and the connectivity of the power graph and the reduced power graph and enhanced power graph. In this paper, we also list the open problems. What are the problems which are still unsolved, or we have also give the references for which what the problems are not still solved. So, and there are in this article, in this paper, authors prove the results for abelian groups, non-abelian groups like dihedral, quaternion, alternating, symmetric groups, cyclic groups. They also gives vertex connectivity and L spectrum of the
Okay,
reduced power
thank you,
graph and
ma'am. Thank you so much
enhanced
for your
power graph.
presentation. Actually, we are running the short of time, so that's why I'm concluding it before. Thank you so much for the
Th-
presentation. You can
Thank,
leave.
thank you, ma'am. Thank you, ma'am.
आप इन्हीं को करेंगे फिर आपसे मेरी
कंटेंट का लिखना ना कि किस कंटेंट पे कौन सा
ठीक है
अभी आप ऐसे तो तो होने के लिए सामने खड़ा होना
नेक्स्ट विक्की है क्या विक्की
नॉट निशा
निशा ठीक है निशा को करते हैं ऐड करो इसके स्लाइड भी डाउनलोड कर लो
मंजू भी हो गई थी मंजू भी हटा दो शायद अब मंजू का ही हुआ कंप्लीट अपने आप हो जाएगा
So I would like to invite our next presentee, Nisha from the physics department. Her topic of presentation is analyzing the structure and catalytic activity of nanostructured ferric.
Ma'am, am I audible to you?
Yes, ma'am, you are audible. Just give us 2 minutes.
Hello?
Just we are processing with your slides.
क्या नाम निशा फिर तो हो ही जाएगा फिर
समझ रहे ना इसमें फिर ये जाती जैसे वो मेरे फील्ड से रिलेटेड तो मुझे ये पता है किस फील्ड पे क्या बराबर इंटरेस्ट उसमें चलना
ना ना मैं चिड़ू क्या था चिड़ू वाली कुछ नहीं कहूँगा
वाले बता
Ma'am, please open your front camera for the video description. Ma'am, are you there?
Audible to you? I'm already in the meeting.
Okay, ma'am. Ma'am, please, we cannot hear you. Can you speak little louder?
Yes, ma'am. Hello?
Okay, ma'am, you can start your presentation. You have 6 minutes timing, ma'am. You can wind up
Sure,
your presentation within 6 minutes. Thank you.
Thank you, ma'am. Good evening, everyone. I am Nisha from Physics Department, going to share my presentation on the topic of analysis of the structure and
Ma'am, we
analytical.
cannot see you. We cannot see you. Please open your camera, your front camera.
Ma'am, will you please share my
And try to speak little louder, ma'am.
Will you please share my PPT on your screen?
Yes, ma'am. It is shared only.
It is on the screen itself.
It is on the screen itself. Can you see your
Sorry, it's
slides?
not. No.
Actually, your the PowerPoint is on the screen itself from our end. If you can see the PowerPoint, are you able to see the PowerPoint?
So now now I'm able to see it.
So, are you able to see it now?
Yes, sir.
Okay, so you have like 5 minutes with you. Please compile with the topic, and the presentation
Okay,
is in
sir.
front of you. Please compile. Hurry up.
So basically, I have in the introduction part, I have put on like the iron oxides have well established electrical properties for many reactions such as the decomposition of alcohol, selective oxidation of carbon monoxide,
Can you
decomposition
please speak
of
a bit louder?
hydrogen
We are not able to
peroxide,
listen to you.
and discoloration. Yes, sir. Discoloration of synthetic dyes and have been extensively studied for environmental application. The basic application for which I have searched on the nanoferrite structure is basically it is going to be used in the cases of the MRIs and the anti-cancer.
Ma'am, your voice is not at all clear. We cannot hear you. Even we can't see you.
Ma'am, just check it out.
Over.
Is it audible now, ma'am? The nanostructure spinal ferrite were synthesized by the co-precipitation and the characterizing various ways to understand their structure and use them as the catalyst. Basically, the work of the catalyst is that is going to increase the rate of the reaction. So we have seen by doping such kind of
Okay, thank you for the presentation, ma'am. We have our judges. They are having few questions related to your presentation.
Sure, ma'am.
Hello.
Yes,
sir. Nisha, can you hear me?
Yes, sir.
Hello. Okay. Why we study materials on nanoscale?
So nanoscales is basically just to find out, like in the nanotechnology, we have seen that the study of the nano particles are somehow going to make elegant and smart nanoparticles. In the biomedical implementation of the nanoferrites, including the cell signals, hypothermal and magnetic resonance imaging, just like the MRIs, are going to be
Can you
made
speak louder, please?
Yes, sir. Anti-cancer function treatment we can do by it. Nanorobots we can make it. Drug deliveries we can do with the help of these structures. And the anti-fungi or the antibacteria, biosensor and
Okay,
the brain
thank you so much, ma'am, for your presentation. Thank you.
Thank you, ma'am.
You may leave, ma'am. Thank you so much for your presentation.
Okay, ma'am. Thank you.
Reconnect me.
I think there is some technical issue.
Thank you so much to our co-faculty members, Dean in charge, and I would like to thank Dr. Renu Sharma ma'am for choosing me as a moderator. Thank you so much, ma'am, and thank you everyone for maintaining the patience and encouraging the research-oriented program. Thank you so much.
Excuse me all. Yeah, before you leave, 1 minute, please. Please mark the attendance and the leaving time of the attendance, please. Thank you very much.
Yeah.